time. Okay, all right. Describe the connection between the two areas via the arcuate fasciculus, and then the concept, which is still intriguing. There's this other node that connects with both areas, and you'll see we still figure out that that's the case. And then the model got a little better after Geschwin added more to it, talked about the reading and writing, the role of the occipital lobe, of the auditory cortex, motor cortex. So as you see, the language network became more and more complicated. And so, but everything was kind of based on the, the aphasic syndromes I learned. Those 7, 8, 9 aphasic syndromes were all stroke-based. And I really like this study. It was right after I finished my fellowship in aphasia that this paper came out, where Nina Dronkers scanned the brain of the patient, Broca's patient. And, on the surface, you see there is a lesion in the Broca's area, in the inferior frontal gyrus. But then when you look at the slices, you see there's a lot of white matter damage also. So what all we attributed to the Broca's area in terms of processing of phonology and syntax, then now you look at there's probably more than that. There's also the connection between the anterior and posterior areas of the brain. And then during the same time, in 2000s, we got to learn more about the tractography, the tractographic organization of language, with the arcuate fasciculus, the external capsule, the uncinate fasciculus, the Aslan tract, which connects the dorsal frontal to the Broca's area. And this was the model that I started working with, the Friederici's model. And it's, as you see, mostly it's the perisylvian area, not much the dorsal aspects of the frontal lobe. And as you see, not just the posterior temporal area, superior temporal area, but middle as well is involved. And not just arcuate fasciculus, but also other dorsal fasciculi also involved with the language processing. So my exploration of the language network started with the functional imaging. A lot of know. For those of you who might not know how it works, when there's an increased metabolic activity in the brain, in that area there's more consumption of oxygen, so there's more oxygenated hemoglobin, which changes the magnetic field of that area, and then that creates a signal, which is the bold signal. This is per Ogawa in 1990, and that's how we started to look at the functions of the brain and different cognitive functions. Hundreds of papers came out after that. It was a boom in our understanding that we could actually see what's happening in the brain. And I'm having difficulty advancing my slides. I don't know why. Okay. And then, as Swati said, I got very interested in the hemodynamic function because it's abnormal in stroke. We were missing activations because of that. So this is just to show how it works. Each time, this is a naming task. When the person sees an object and names it, you see a increase in hemodynamic function. It goes down, and then we present it. This is a very long trial, 20 seconds between each trial, because we wanted to see exactly what happens after the peak. And then when you average them, you can see the picture on the right side, which is the occipital lobe and then the perisylvian areas involved in the processing of language. So that's how we figure out how things are going. In the beginning, we started looking at nouns and verbs. We thought we could find a distinction between them. On the left side top here, we could not. Then instead of just looking at nouns and verbs, we looked at verbs with different syntactic information, so-called the argument structure. And then we found that the more arguments that are associated with the verb, we were getting this activation in the angular gyrus bilaterally more on the left side But then we still couldn't believe it at that time. This is 2004-5, because we still thought everything syntax is in the Broca's area. And that's why I showed you that scan from, at that time, we had not seen the scan, the Menina Drunkard scan, right? But when we did it in 2 data sets, younger people and then people who were in their 50s average, we saw this is very robust. And then the argument structure information is just there. So some of the earlier, planning for grammar is happening actually in the posterior areas of the brain. And then later, this was with my work with Cindy Thompson, and then Aya Meltzer moved this forward, and then they came up with this map for the syntactic processing involving posterior, superior, and middle temporal gyri, angular gyrus, and Broca's area as the areas involved in syntax. So this was all based on the stroke aphasia literature. And my quick journey today for where syntax is processed. Then I got interested in the semantic aspects, and that's because in primary progressive aphasia, you have this opportunity to look at that, because in strokes, if you get a cerebral artery stroke, and then you get a lesion in the anterior temporal area, it's not as clean because you may have some frontal damage, you may have some parietal damage. So it's difficult to understand the correlation between the lesion and the symptom. But in primary progressive aphasia, actually, we can see more about that correlation, as I will show you. And at the time, resting state became more and more popular because we didn't have to have a task. And for patients with aphasia, that would be easier. And in resting state, there are a few ways we can look at the function of the language network. One is the node-based, when you pick your regions of interest and look at their connectivity. Or you can have the seed-based, where you pick a seed and then look at the connectivity with the rest of the brain. Or you can do what I think is not here is the independent component analysis, where you pick every single voxel and ask the question of what other voxels in the brain it's connected to, and then you come up with different components. And at that time, in 2013, this paper got published where Marcel Mesulam, my then mentor, my colleague now, he looked into individuals with semantic primary progressive aphasia. These are people who have difficulty with naming, severe difficulty with naming, and also understanding words. So it's called two-way naming deficit. And he figures that the correlation between word comprehension and atrophy is really lateralized to the left, to the temporal pole on the left side. And that kind of tells you, okay, so it involves in naming, it involves in understanding of names. And at that time, the temporal pole was and was not part of the language network. And the question was, is it or not? So there were some issues figuring it out with functional connectivity. So what I did was, I said, okay, well, we'll just put all these equidistant nodes in the brain, and we're going to just do a data-driven approach and then see how these are connected. And are the known regions, the posterior temporal, the frontal, are they connected to the temporal pole? And as you see, so this shows the connectivity between all of these nodes. The ones that are more connected are in darker color. And then you see what pops out here are these regions. So you have the posterior temporal, which is equivalent to the Wernicke's area. You have the Broca's area, you have the angular gyrus, which I showed you earlier, and then you see the temporal pole. So we figured that, yes, the temporal pole is actually connected to the nodes of the language network. And then we looked at left side and right side, and we figured the connection is stronger on the left side. And that's because these guys are all right-handers. So this is because of the language component. I also did a independent component analysis with help of Michael Grecius at Stanford. That's the lower image here. And then you see if it without even any presumptions about the seat, if you just look at the voxel-wise analysis of the whole brain, you see the left side comes up very much stronger. So that seems to be the language network, but wait a minute. So what I looked at in the beginning with the Federici's model, it was all perisylvian. So all these areas now are showing up. Are they really part of the language network? So that was what we found with the temporal lobe. So I did the same thing with the premotor and prefrontal areas. And then you see the same thing. It's more left lateralized. So the whole area is involved, connected to the main nodes of the language, and then you have this asymmetry altogether. Okay, so this is where we were in terms of in healthy individuals, how the organization is. We came up with this 3 or 4 node model. First, the 3-node model, and then we wanted to see what happens in PPA in terms of connectivity. And before I talk to you about PPA, I thought I'll tell you a little bit about the language evaluation, how things may be a little different between what we did in stroke and in PPA. So in stroke, everybody who comes to you in your clinic and they can't communicate, you want to know if it's motor speech disorder, is it aphasia? So you do a naming battery, you ask them to describe a picture, and then based on the fluency and then the naming situation, you can find out if you have non-fluent or fluent aphasia. And then you also take into account comprehension and repetition. And then you come up with those syndromes that you all know very well. In PPA, we do almost the same thing. So these are the tests we use in our research at Northwestern. Boston naming for object naming, and then the wall picture description instead of the cookie theft. There's also the Cinderella story where they describe the story and then it gets analyzed in more detail. And the only thing that's probably added is the Peabody picture vocabulary test, which is the word comprehension. And I'll show it to you later. And the repetition again, but we use the law of repetition. And then for grammar, we use these 2 tests. Both of them developed at Northwestern Assessment of Verbs and Sentences, NAVs. The picture here shows you sentence production based on probing. So you tell the person, show me the picture where the cat is being chased by the dog. And then with passive sentences, they have more problem. With active, they have less problem. And then to take out the working memory, verbal working memory aspect of it, Sandy Weintraub and Marcel Muslim, they came up with the idea of Northwestern anagram test, where you have the words in front of you and they're supposed to make sentences based on the picture they say there. The girl is tickling the boy. So and then we use both to come up with a combined score for grammar. And this is the Peabody picture vocabulary test, where people see the word and they're supposed to match it with the picture, and then people with semantic PPA cannot do that because citrus to them doesn't sound like a word. It sounds like a foreign word. They may tell you what's citrus. Because everybody is familiar with the cookie theft, I thought I start with that. And because of what I learned from Nancy and Martin Albert, and then they really focused on that. So this is how we still use it in our clinic. We use R.G. Hillis's version, which is more colorful and more modernized. So this is how a healthy individuals, non-aphasic individual describes. The picture, as you see, a mother and 2 kids in the kitchen. The mother is washing dishes. So long sentences, embedded clauses. And what happens is they come up with 18 content units, a range of 13 to 24, and 4 to 5 words per utterance. And then I was able to find, actually, if anybody looked at it for PPA, and I was able to find the Marshall study. So let's listen to the 3 subtypes of PPA. And then this is evolving. There are now more subtypes, so we'll just focus on these 3 just for you to hear them. For those of you who don't know PPA, I know some of you do know PPA. So this is the agrammatic version or variant. And then you see what you see on the left side is the atrophy map. And they have mostly atrophy in the frontal area of the brain. And it's mostly caused by frontotemporal degeneration. And either of the tau or the TDP type pathology. And this is how they sound like.
Can you tell me what's happening in the picture?
Obviously, the man, a boy, and a cookie is on them, and his daughter, a sister, and when she's gonna throw in with them, and they've broken a book on the store, on around one of them. A lady, and she was having washing on them, and there were taps on them, and drain. It's come on the floor, one there, horrible, a tile one there, and this man out there, bear, maybe going in the winter on there, working out the window. Maybe this little girl looking so on there. I don't right on there.
Okay. So does that sound like Broca's aphasia to you? So you see, depending on where the atrophy is, the phenotype is different. You know, a lot of people with Broca's stroke aphasia, they do have where the stroke is, because it disconnects the Broca's area to the superior frontal areas where the motor planning is. They do have more non-fluent speech. And that's the Aslan tract, right? We do have PPA individuals who do have that non-fluency and the apraxia of speech. And we have people who don't, like this person. She definitely is agrammatic. Her sentences are small, but she has a lot of fillers. And her articulation is actually not that bad, right? So we do see this kind of agrammatic patients with PPA. And I'm not sure, I mean, I don't see as many stroke patients anymore. So you got to tell me if you see similar. You might see the same syndrome in, you know, maybe some embolic strokes as well, which they have the agrammatism, but not the apraxia component. With the grammar, because the grammar is pretty bad here too. And then the conduction, you do have the phonological dysarthrias, but this person doesn't. Yeah. So conduction would look more like this one. And then see, these all can overlap. It's not that easy. So this is an individual with logopenic type. And then you're going to hear again, non-fluent speech, but this time it's because of the pauses. They can't get to the words. They do access the words. It's not like the semantic individuals who can't get to the word at all, but they just can't get it out because of the atrophies right here at the temporoparietal junction where the planned word can't get to the Broca's area. And so they do have actually phonological difficulties too. I don't remember if this person does or not, but let's see.
When you're ready, what do you see happening in that picture?
I see a mum washing, drying some plates, with water running down. I see a young boy in the other part of the trying getting off or nearly getting off the stool with his sister below.
Perfect.
No, it wasn't. It was awful.
Very frustrated. So you see how that sounds, a lot of pauses. And that's the type of word finding difficulty seen in Alzheimer's disease as well. But in this specific phenotype, they don't have trouble with anything else but language. They have good memory, they can do their activities of daily living. You see the atrophies mostly in the posterior temporoparietal area. And this is mostly caused by Alzheimer's disease, but not 100%. You can sometimes see other pathologies too. And then this is how the semantic aphasic person will sound like. They have the atrophy in the left and right-handers, left temporal pole.
What's going on in this picture here?
Oh. Well, there was a woman and her children, and it's a house, I suppose, and there's a little window from there, and I don't quite know what that is going down precisely. And then the children are Cooney jar. I don't understand that quite either, what they're doing. And gosh, that one's foot is about to come off. Oh dear.
That's very good. And what's happening over this side of the picture? Is anything going on out here?
Well, there's a people all down the end, I suppose. Just that way, or maybe not. Maybe it's buildings, maybe it's trees, etc. I don't know.
That's great. That's excellent.
You don't see that disfluency that you see in the logopenics. Logopenics are also, they can get to the words, but it's completely different. You have very fluent output, good grammar, but then the semantic type is the one that they tell you, I don't know what that is. And then that's very typical of semantic primary progressive aphasia. Or you have, semantic paraphasia, that leg is about to come off. I think he's talking about the stool coming, falling. So these are, and then when you talk to them, and then in the beginning when I didn't quite know how things were, and I was interviewing them for our research, and saying, do you have high blood pressure? And then the person would look at me and say, what's pressure? And they were kind of offended that I'm saying something that they don't understand. And then they can come up with long sentences and then discuss sometimes very difficult concepts. So, okay, so now you kind of saw where we were with the language network in terms of the functional MRI. I showed you some of the atrophy patterns, and then we wanted to know how things are in PPA. With PPA, the problem is, you have the atrophy. So if I say, okay, there's decreased functional connectivity between two areas, then you're going to tell me, well, it's because there's atrophy. So we had to kind of get around that. And so we did have a limited number of patients in our cohort who did not have atrophy, at least based on the threshold we were applying. So we thought those were actually perfect to look at connectivity. And then after that, we looked at the ones with atrophy. We want to look to see what the pattern is in each of the PPA syndromes. And, the idea is to, in Swati's lab, you're doing a lot of prediction type studies. You want to know target engagement when you're doing the treatment. Are you engaging the right network? And then is lesion in some part of the network affect responsiveness to treatment? And then you want to use this information for neuromodulation if possible, right? For therapeutic interventions. So these were the 10 individuals we had with, and all of them had non-fluent primary progressive aphasia. Oh, I'm sorry, I didn't know you were seeing it. You are seeing my screen. Okay. Okay. Can I close this one? Yeah. Is that, there's a way to. There was a way to just get rid of this. I don't remember. Okay. All right. So these were the 10 individuals who had the agrammatic type PPA, and they had no atrophy. So we said, okay, well, why don't we just first look at those two main language nodes, IFG and MTG. We didn't look at temporal, anterior-temporal lobe because we knew it's not relevant for grammar, right? And then we picked two areas from the default network. And the default network is the biggest resting state network in the brain, you know that. And you know the major seeds of it are the posterior cingulate and then the mesial prefrontal cortex. So they didn't have atrophy in the language areas, they didn't have atrophy in the default network. And then when we looked at all these patients, there was a little bit of decrease in connectivity in PPA, no decreased connectivity in default network, but it was not quite significant. But when we looked at the, oh, I'm so sorry. This is the atrophy maps. These are the atrophy maps. So there was no significant difference between the atrophy or between the cortical volume. And then when we looked at the connectivity between the two nodes, you see that in PPA, the connectivity between IFG and MTG is decreased, but in the default network, it wasn't. So that served as the control. So there's definitely decrease in connectivity in the absence of significant atrophy. And then if you did the seed-based analysis, put the seed in the IFG, you see that the connectivity between IFG and the rest of the brain is much decreased. So that kind of tells you what's going on. Before the atrophy, you have this decrease in connectivity. And it looks like this might be a case for the type of pathology that causes non-fluent aphasia, which is mostly the frontotemporal type, either 4R tauopathy is what we call them. And so then we moved on to look at the people who did have atrophy. And then this is just to show you how those tests I showed you earlier look like. You know, they're the combined NAT now is what we call it out of 15. It's very low in, this is the grammar score in agrammatics. In logopenics, you have the naming that is lower and the repetition is also impaired because of that specific area of the brain that is involved in repetition, the temporoparietal junction. And then the semantics, you see that they have the most severe naming problem. Do you see that? The BNT is 9 compared to the other ones. And they have significant word comprehension problem compared to the others. So that's how we categorize them. And then when we looked at the connectivity, so we just started with the simplest idea, just look at these 3 nodes. And then all of them have decreased connectivity between IFG and MTG. And then specifically in the semantics, you have the ventral pathway of language. Remember the ventral and dorsal pathways? The ventral pathway is related to lexical and semantic processing, and then the dorsal phonological and grammar. So, okay, so it looks like we were able to figure out how semantics are different from the G and L subtypes. And then we looked at the correlation with symptoms. And then as you see, the IFG MTG correlated with repetition and grammar, mostly because of the involvement of the dorsal pathway. And the MTG ATL correlated with the naming and word comprehension. See, we were not the first people who looked at the resting state. There were other groups who had looked at resting state changes in PPA, our colleagues, Bill Seeley and colleagues in UCSF. The correlation between the connectivity and symptoms were not quite well known at that time. So I think that was probably what the contribution we had to the literature. Then the question was, okay, so what's the difference between the agrammatics and logopenics? They had both decreased connectivity between IFG and MTG. But if you just put the seed in IFG, that kind of tells you what the difference is. The agromatics have more decreased connectivity in the frontal lobe, all the way up to the top. And then the logopenics don't. And well, semantics, of course, they behave differently. So I think looks like that was where we should look at. So we put seeds now in the temporoparietal junction. Our reviewer said, why don't you put just seeds in also the posterior part of the of the superior temporal gyrus. We did that. And then what really defines or separates these two subtypes is the connectivity between Broca's area and superior frontal area in agromatics, and Broca's area and the temporoparietal junction. So that's the distinction between the two. So with 3 nodes, we couldn't part them, but with more than 3, we could. So see, as we are trying to figure out, you see what areas are really involved in language. It's just the 3-node approach was too simplistic. We had to add the temporoparietal junction, and we had to add the superior frontal area. Later, and then more recently, we had the opportunity to look at FDG PET, fluorodeoxyglucose, look at the metabolic activity. And with the resting state, if you only look at the amplitude of fluctuations of blood flow, what's called the ALF, low frequency fluctuations, you can get the same. So we found the same pattern as in PET in both of them. And then that shows you where the metabolic problem is. And these results are all corrected for atrophy, by the way, because as I said to you, the atrophy is always on the way. So when we put that as a covariate, that takes care of it. So what you see here is what's beyond the atrophy. So now you see in agromatics, where we had IFG, MTG decrease, it was really driven by IFG. We still are analyzing the data, so we will have to show it in numbers too, but that seems to be the case. And in the logopenics, it's the other way around. It's because they have temporoparietal junction and temporal hypometabolism. So now we are kind of where we are is, we want to do multimodal analysis to see how much each of these contribute to symptoms, the atrophy, resting state, and PET scan. That's going to be very helpful for our understanding of symptoms and then also responsiveness to treatment. Okay. Any questions so far?
We'll wait for the end.
Okay, we'll wait until the end. So, why are we doing all this? We want to help patients, right? And then as, you're very aware, one thing that we can do to see if, for example, atrophy or decreased connectivity in any of these areas would affect responsiveness to treatment. When I was in Arizona, I worked with Maya Hendry and Paige Beeson. They came up at that time, it was 2008-9. They came up with treatment for PPA. Before that, there was not much going on, maybe 1 or 2 case studies, because people thought if it's getting worse over time, because PPA patients can get worse, it's degenerative disease, and they can have trouble with other modalities too. So why bother? But that's not the case. You can actually help them recover some of their problem, and then if they get worse, you can do it again. So you do have to do multiple interventions. And then what Maya did, she looked. So this is what the map that I had. This is the map for agromatics, right? And then she looked to see if atrophy in anywhere in this map would correlate with responsive to treatment. And then she found out that atrophy within posterior middle temporal gyrus and inferior would correlate with the responsiveness to the treatment that she called VISTA, video implemented script training in aphasia. This is the left picture. I tried to look at her data, the treatment data pre and post at the time. So this is actually before the pandemic. We need to get back to this. Pandemic kind of fell everything apart. We didn't have enough patients at the time to look at connectivity the way I looked in single subjects or in the cross-sectional study. So I looked at amplitude of low frequency fluctuation, and you see it's decreased in PPA patients. This is the top picture. And the lower picture shows you what happens after treatment. So we had increase in amplitude of low frequency fluctuation. That's really equivalent to the metabolic activity in PET. So it looks like we showed some target engagement. That's exactly where you want to see it, right? Temporoparietal junction, you have the Broca's on the left and right. So this is to be continued. We need to analyze the rest of the data. And then the other goal that I had, and I came to Boston for that, to learn how to do TMS, was to use this kind of information for therapeutic modulations. And with transcranial magnetic stimulation, you try to either stimulate or inhibit areas that are contributing, are not activating enough or activating too much. So we were on the path to do that. Again, this was interrupted by the pandemic. This is where we got. We were trying to figure out if we can find an area in the parietal area that would tap into the language network and also in the memory, because we wanted to see how that works with people with dementia and aphasia. And then you see that the more posterior you go, your modulation taps into the hippocampus, and the more anterior into IFG. So if you have a patient with dementia and aphasia, then you can use these different areas to modulate their networks. So as it turned out, the way it turned out, we're going to actually continue this, but with transcranial electrical stimulation. So hopefully at some point I'll be back and then report on that kind of study we are doing. We are in the process of writing the IRB and all that. Okay, so I talked a lot, so I have time to summarize everything. You saw the whole thing with the language network. It's an evolving thing. While we were working on PPA, a beautiful paper came out from Machin and Hickok. And so they looked at syntax, so MTG, the role of MTG and comprehension. I mean, it's kind of equivalent of the Wernicke's area that was known. But then, as you saw in agrammatics, it's also the connectivity is abnormal. So that region seems to have a dual function. It taps into both and the ventral and the dorsal pathways. And then you see the role of ATL and angular gyrus. And all these, so these are mostly the linguistic components. And then in brain resting state or functional map, you have these gradients where you have a small one and then you have one that goes on top of it. So this is how things are structured in the brain. So the ones that are more ventral, they go around, and then you see that the yellow here, which is the morphosyntax, right? The one that is inside it, these blue ones, are mostly related to articulation and phonology. So the one that's inside the core is articulation, phonological, and then the one that goes around it is both comprehension and syntactic production. So that's where we are. And then you see this, superior frontal area, which Hickok and Popo had mentioned it before, but some other models didn't, right? But as you saw, we had to include it to kind of be able to understand what's happening in PPA. So we are learning about the language network through PPA as well. And another beautiful study, and then, F. Fedorenko, of course, here in Boston has been doing beautiful work on localization of language. And you see, this is her meta-analysis. And you see all those areas are involved. And I learned from people in your lab, Swati, that you're looking at the role of executive function. How much of that frontal area is executive, and then how much of it is also related to social cognition, because there's that too. So that's where we are with the evolving situation with the language network. And then just to summarize, so you saw that in people who have less amount of atrophy, you can have decreased connectivity. That sounds like this is a leading indicator. Decline in word comprehension, we found that it was because of the decreased connectivity in the posterior and anterior temporal regions. But FDG PET showed us that it's really the anterior temporal that's driving it. And the same thing with syntax. We had more temporofrontal areas, but then it's the frontal area based on our, both the atrophy maps and PET results. It's the frontal that seems to be driving it. And then I showed you some examples of how this kind of information can be used for target engagement, for responsiveness to treatment, and for hopefully neuromodulatory interventions. Again, you know, they are very interesting studies going on at Penn, at MGH, and Hopkins, mostly reading, writing. I know Kirana is working on it in Toronto. So we're all working on this, and then we'll see. Hopefully we can combine it, all of our studies as a network, and then understand more, and then help individuals with aphasia, progressive aphasia more. And I have to thank all the people who worked with me during the past many years, all the talented students who are now, some of them are doctors, some of them are data scientists, some are still working with me, and some of our faculty, and you know, Amy is in Mayo Clinic now, may actually come back to Northwestern, so we'll see. And many, many others at Northwestern, and other institutions at Stanford, Austin, University of Texas, Austin, Harvard, UCSF, and others, and our funding mechanisms. Thank you so much.
Fabulous, perfect time. We have some time for questions. Because this is hybrid Borna, it's always complicated to see who can.
There are some texts.
I will watch the questions online, but let's start with the questions in the audience here first, and then we can tell.
Sure.
I'll keep track of the chat. But let's start here first.
So I think it was your 2019 study, you showed sort of like correlations with language assessments for the 3 groups of PPA subtypes. And I noticed that your agrammatic subjects were maybe approximately double the amount of recruitment for the logopenic and the semantic variant. And
so we have 36 agrammatics, 20 logopenics.
Yeah, and then I think it was 17 maybe for semantics.
Semantics, right.
So I was curious, in your professional experience, is that sort of like, is that agrammatic is more common, or maybe does it have something to do with severity and getting people into research studies?
Yeah, I think, you know, that's a great question. You know, because at Northwestern, we really like syntax. We are very good at picking up on the grammatism. So so there is some differences between different centers. There really shouldn't be. And so we have more agrammatics than others because we pick up on more of them. And with the, you know, as you saw, you know, we have 2 tests. And however, not because some of the, you know, patients that you may put it in the agrammatism, the agrammatic group, they could be logopenic, because some of the, you know, there's a paper by Sujin Cho and Cindy Thompson who showed logopenics do it because, you know, it's not just the front, it's the whole network. They have some agrammatism too, but we are very careful not to include those. And then the other thing is we use only the phenotype to put people in different groups. We don't look at the imaging. Some other teams, they look at the imaging and say, oh, this person is agrammatic, but because they had atrophy in the temporoparietal junction, we put them in the logopenic group. We don't do that. We just go by the phenotype because we believe there is a separation of powers between the linguistics and then what you see in neuroimaging. So some of the agrammatics are actually the Alzheimer pathology patients. Did I answer your question?
Yes.
Yeah.
I'm watching. Can I ask a question as I'm watching the chat? In the TDC, the TMS, the stimulation study, you pick parietal, and so you don't have to go back to the slide, but I'm just wondering what's the rationale for picking that region as a stimulation?
Well, this was
site?
because of our collaboration with Joel Voss, and Joel was really interested in memory. And then, you know, you can't get to the hippocampus from the surface. So he found this very nice area of the parietal lobe that would tap into the hippocampus. And then for language, it's the same thing too. You know that the temporoparietal junction can tap both to the ventral area and the dorsal. In that case, what they found mostly tapped into the IFG, as you saw.
So it doesn't matter that there is no atrophy in the parietal region or more. It's not. It has nothing to do with whether it's damaged or not. It just
Right. Great question. Great question. So in neurodegenerative disease, the difference between atrophy and lesion that you can see in stroke is there are still viable cells there. If you remember, there was a paper by Santi, 2003. That was probably one of the first fMRI studies of PPA. And then she showed activation, actually, that was a task-based fMRI. She showed activation within the areas that were atrophic. So that tells you atrophy actually doesn't even mean that neurons are dead. Sometimes it's the supporting tissue, the glial cells. So, and then that what makes it fascinating, because you can still modulate the areas within the atrophy. Again, depending on how bad it is. Like in some of the semantic patients, the atrophy is so bad that there's not much left. So it depends on the severity. Yeah.
Hey, Dirk has a question for you, so maybe you can read it aloud. Or
Yes. Right number of newly distinguished PPA subtypes. Yeah. So hi, Dirk. You know, it's getting more and more complicated because, as you know, the way that we were approaching the symptoms with the 20% of, or probably even more, we couldn't put them in each of the different bins. So now there's more and more subtypes. It's becoming more and more granular. So what we do, and then the question is, how can it, you know, Dirk is asking the great question, that how would it affect patient? How would it affect patient? Mostly rehab, right? We really don't care what they're called. So what we do now, what Emily Rogalski does or what Maya Hendry has done, you just go with symptoms. Do they have more difficulty with naming? Do they have more difficulty with production? And you just focus on the treatment rather than what kind of subtype it is, because the subtypes matters really that, you know, clinically for me to think about what's causing it. For example, if I see a semantic person, aphasic person, and I see a huge atrophy on the left side, I know it's probably frontotemporal degeneration of the TDP type C, and then almost nothing else can do that. So I don't send them for a spinal fluid exam to look at Alzheimer's. And that's important for our clinical trials, because now that more Alzheimer's medications are becoming available, that helps us with treatment. So in terms of treatment, there's a pathology treatment with drugs that, you know, you're relying on, you know, your amyloid scan and a spinal fluid. And then there's the symptomatic intervention, which is, you know, we rely on speech pathologists. So that we just focus on the deficit. However, of course, you know, the deficit in semantics, the naming deficit is different than people with logopenics. So that's really how we get around it. Otherwise, I think it's going to get more complicated with all the developing subtypes. It's very complicated. They do. And they sometimes get confused because, see, I mean, if you tell somebody you have PPA, they think that's the diagnosis, but that's really the clinical syndrome. PPA can be caused by frontotemporal degeneration or Alzheimer's disease. So the drug that you use is different. And then they say, oh, yeah, so I have PPA, but I have Alzheimer's disease. And then, you know, I have to really tell them, sit down and talk to them. Say, I see Alzheimer's, 90% of the time it's memory, but 10%, it could be language, or it could be vision, or it could be behavior, or it could be executive function. So we really have layers of diagnosis: clinical, imaging, and pathologic. But yes, we have to, it's very complicated. And then with some of the agrammatics, they can get worse and they develop Parkinsonism, they may fall, they may have gaze palsy in the PSP type, progressive supranuclear palsy. So you tell them, oh, you have PSP, and then they ask you, oh, so I don't have PPA? And then I have to really sit down and talk to them. Now you do have PPA, but now with PSP features. So now the nomenclature and the whole description of these syndromes are very complicated. There's a whole committee now assigned For the nomenclature, yeah, because it's gone very complex.
Okay.
All right, thank you so much.